Every HLA allele your immune system carries is a trade-off. The same molecule that helps your T cells kill a tumor might also drive autoimmune disease. I asked a simple question: is there a single axis of HLA immune potency that's good against cancer but risky for autoimmunity, and can we measure it from sequence alone? So I built one. I call it engageability, how permissively an allele's TCR-facing surface emits diverse T cell receptors. I scored 676 common class 1 alleles from crystal structures, recovering the known TCR contact residues exactly. And, critically, it's orthogonal to HLA evolutionary divergence, the established breadth metric, a genuinely new, independent dimension. That changes the picture. Run through the same cancer analysis, breadth and engageability point in opposite directions. Add presentation capacity and CD8 engagement, and potency stops being a single number. It's a multi-axis decomposition, several independent immunological dials, not just one. Here's where I was rigorous instead of lucky. The germline trade-off, tested directly, came back null, but we can prove why. Mathematically, regressing outcome on potency cannot separate immune escape from immune killing. The arms are non-identifiable. And on public data, the effective sample size is about 8 independent points. So the pre-registered power analysis never reaches 80%. The null isn't a finding, it's simply the data telling us it can't answer this question. So instead, I followed the statistical power to where the data is deep, somatic immune escape across 50,000 tumors using the MSK Impact 50K cohort. Our pipeline first recovers the known control, loss of function selection on beta-2 microglobulin, with a built-in negative control passing exactly as it should. Then the new result. Across the HLA-B locus, we find a robust loss and retention hierarchy, and at its loss-prone extreme, one allele stands out. B5801 is preferentially lost at a fraction of 0.67 with a false discovery rate of 0.003. It survives a 4-check adversarial review. It's not a mapping artifact, not haplotype driven, and not in the MSK Impact 50K cohort's own paper. And none of it was cherry-picked. Every arm was pre-registered. The pipeline caught its own false positives 3 separate times and retracted them on honest controls. Every number traces to fetch primary data. This is disciplined, reproducible science with Claude. And it points somewhere. HLA loss is how tumors escape, so the allele a tumor refuses to delete is one it can't easily flee, a durable restriction element for TCR-based therapy. The alleles it does delete flag the peptides it fears most, so the next step writes itself, and it's one we can take straight to the bench. Map what B5801 and the retained alleles present and prioritize the escape-proof targets. A new axis of immune potency, proof of what public data can answer, and a reproducible lead to build the next therapy on. That's the work.